So usually the exergonic reaction and endogonic reaction, they occur at the same time, because the exergonic reaction releases heat, that's why it is hot, and the endogonic reaction absorbs energy, absorbs heat, so it is cold. And when we put a hot reaction and cold reaction together, then it will fit the first law of thermodynamics. Energy is transferred, cannot be created or destroyed, but energy can only be transferred. So the heat released by the exergonic reaction will be transferred to the endogonic reaction. An example will be this. ATP, when we break the bond between the phosphate group, then the covalent bond between the phosphate group will be released. So this reaction, ATP into ADP and inorganic phosphate, it will be the exergonic reaction. The heat that is released, or the energy that is released by breaking the phosphate group, by breaking the covalent bond between the phosphate group, will be used to drive this reaction, reactant A and B, into product C and D. In other words, this reaction at the bottom, this is an endogonic reaction, and this reaction on top will be the exergonic reaction. So this is something that you will learn again in anatomy, and you will learn it again in physiology. It's the muscle contraction. We will just very briefly to talk about it. So what happens is that when your muscle relaxes, you require ATP. This is a little bit counterintuitive. You may think that, okay, we need energy to do work, to contract muscle. When we relax, we do not need energy, but this is actually contradict to our intuition. When we relax our muscle, when our muscle relaxes, we require an ATP. When ATP binds to our muscle, we have two types of muscle fiber. One is called actin, thin filament, actin. And then the other one is called thick filament, we call it myosin. So when ATP binds to the myosin, then our muscles start to relax. And then the myosin, it will hydrolyze ATP into ADP and inorganic phosphate group. Then the detachment of the ADP and inorganic phosphate group will cause the bending of the myosin. When myosin is bent, then you will move relative on the actin filament. When myosin is moved relative on the actin filament, then you will shorten our muscle, and that's why we contract when ADP and inorganic phosphate group is detached. And then what happens is that these cycles just repeat after it is moved or shortened, or after the myosin is moved relative on the actin. What happens is that ATP will bind again on the myosin, and then as ATP bind, then myosin will be detached from actin. When myosin is detached from actin, then muscle will be relaxed. And then we can start another cycle. The question is, what if we do not have ATP? What if we do not have ATP, what will happen to the muscle? Think about this. When we do not have ATP, what will happen to the muscle? Muscle will, as I said, that binding of ATP is the first step, is to relax the muscle. Because muscle is contracted in the previous step. So if there's no ATP bind here, then the muscle cannot relax. The myosin cannot detach from actin, the muscle cannot relax. That means the muscle will be contracted continuously. And may I ask, when will we not make ATP? When will when we are dead. When we are dead, we stop making ATP. In other words, when we are dead, our muscle will be contracted, remain contracted. What do we call this phenomenon? This phenomenon is called rigor mortis. That is the exact reason why dead body, they are so stiff, because dead body do not make ATP, and when there's no ATP, we cannot relax the muscle. The myosin will remain attached to the muscle as if the muscle are contracted. So this is like a real-life application to what we just learned here. Now, metabolic pathways. Metabolic pathway, it's just a series of reactions linked to each other. Reaction in the cell, they do not occur randomly, haphazardly means randomly. There's always a reactant and product, and the reactant and product, they are specific. The main reason is that the energy that we use usually is more efficient if we use it in a small step. It is very uncommon. We invest a large number of ATP in just one reaction. Usually we invest 1 or 2 ATP in a very simple reaction, and then we'll do many steps, many simple reactions to convert a reactant into different product, and then the product will become reactant of different another product in order to get it into the final product. Let me show you a picture to explain what I just said. Oh, it's not here. Okay, this is the picture I want to show you. But we talk about enzyme first. Let me continue here. Okay, let me finish the story. So, usually in a metabolic pathway in our living cell, you have this long sequence of pathway, we call it reactions. So, the product of a reaction is very likely to be the reactant of another reaction. The advantage of doing this is that each step we only need a little bit of the energy, a little bit of ATP. On the other hand, if you want to convert A directly into G, then what will happen? You will have to invest a lot of ATP to convert A into G. In this case, what will be the possible issue? If your body burns a lot of ATP at one time to try to convert A to G, what will happen? You will probably burn your body. Your body will probably on fire, because a lot of ATP is invested in one single reaction. Then you will probably overheat your body. And besides that, if you invest a lot of energy or ATP in one reaction to jump from A to G, you will also probably lose a lot of heat, lose a lot of energy as well. So it is not that efficient. But if we do it step by step, so each step we only invest a little bit of the ATP, then the heat loss in each step will be very minimal, will be very small. Then you won't overheat your body will not burn. But of course it will take a long time to do it. How does your body compensate this issue? So we don't want to burn our body by converting A to G directly, but we don't want to take a long time to do all this reaction. So what can our body do? Think about this. Our body makes use of something called enzyme. Enzyme is a catalyst. What is catalyst? Catalyst is substances that can speed up a chemical reaction. So this is the definition. We give you an example called ribozyme. Ribozyme is an RNA that can act as a catalyst. What does it mean? Ribozyme is an RNA enzyme. Ribozyme is basically RNA enzyme. scientists propose that the first genetic material that occur in a prokaryotic cell, the first genetic material that occur in a prokaryotic cell, they are not DNA. Yeah. The first genetic material that occur in primitive prokaryotic cell was not DNA. They believe it is something called ribozyme. It is a riboenzyme. So it is an enzyme, but it is also an RNA. In other words, RNA and DNA, which one is more primitive? Which one occurred first in our history? It is RNA. RNA is more primitive. It occurs in history first. So, but before RNA, we have ribozyme. It is a RNA enzyme. So we believe that this riboenzyme is coming from a protein or chain of amino acid. And then eventually, so in other words, riboenzyme is like the intermediate step in between amino acid and RNA. So one of the major properties of enzyme is that it will speed up a chemical reaction, but it will not be used up by the reaction, meaning that we can recycle it. We can reuse the enzyme again and again to speed up the same chemical reaction. It does not cause the reaction to go forward, meaning that it does not supply energy to start the reaction. It does not supply energy to trigger the reaction. But what it does is that it lowers the free energy. It does not provide energy to start a reaction because what enzyme does is that it lowers something called activation energy in order to speed up the reaction. So let's go back here. Just a brief review. In order to form product G from reactant A, we do not go from A to G directly because it will require a lot of ATP, a lot of energy at one time. When this happens, our body will be overheat and will burn ourselves. But we use small steps. Each step we only require a little bit of the ATP so that it will generate a little bit heat in each step. In this case, our body will not be overheated. And then, because we have a lot of steps, how do we speed up the reaction? We use enzyme. And I talk about activation energy or energy of activation. Activation energy, what is that? It is the energy that required for the reactant to form product. Let's put it this way. It's the energy required for the reactant to become product. This is a very good analogy. It's like when you want when you want the wood start to burn, you need a match. You need a trigger. So activation energy is like a trigger. So enzyme is able to lower this activation energy to make it easier to start. When it is easier to start a reaction, what will happen? Well, it will speed up the reaction. And yet, enzyme does not change the end result of reaction. That means enzyme does not change the product. It just make it faster. So this is an important figure to help us understand. Activation energy is basically this hurdle. And so in order to this is reaction, this is a product. In order to convert the reactant into product, we need to overcome this hurdle. We call it activation energy. The differences between the blue curve and the orange curve is that orange curve has a lower or smaller activation energy, and then the blue curve has a higher activation energy. The reason is that the orange curve has enzyme. Enzyme will lower this hurdle so that the reactant will be easier to go through this hurdle to become the product. And another question I want to ask you is that, is this chemical reaction exergonic reaction or endergonic reaction? Is this picture here, is it an exergonic reaction or an endergonic reaction? Well, we can take a look at here. The reactant has more free energy than the product because the reactant is higher than the product. And we look at the whiteboard here, the reactant has more energy than the product. So in other words, this is an exergonic reaction here. This is an exergonic reaction. As the reactant is being converted into product, heat will release. So we also need to introduce a term called substrate. Substrate, you can assume that substrate is very similar to reactant. Substrate is very similar to reactant. And then active site. Active site is actually a pocket in the enzyme. Remember, enzymes, they are basically protein. And which level of structure of protein, they will have a 3D shape that can form an active site. We have primary, secondary, tertiary, and quaternary. Which level of structure of protein will form a 3D shape that can have an active site? The answer to this question is tertiary and quaternary. So this equation indicates the sequence of steps of an enzyme catalyzed reaction. Substrate, you can assume that it may be the reactant. When the substrate binds to the active site of the enzyme, then the substrate and enzyme, they become this enzyme-substrate complex. And then the enzyme will speed up the reaction of converting the substrate into product. And then the product will be detached from the enzyme active site. So the enzyme can go back to react with another substrate to form another enzyme-substrate complex and then to form another product. In other words, you can see that enzyme, they do not change and they are reusable. At the end of the reaction, it is still the same enzyme, but the substrate or the reactant is already converted into product. So remember, so this is a reaction. So is substrate is being broken down into the smaller product. We call this degradation. Do you remember an example of the degradation? For example, what we talk about, carbohydrate starch is being broken down into glucose. And then it is an example of degradation. And what do we call this chemical reaction? Starch is broken down into glucose. Other than degradation, we call it a hydrolysis, right? Remember? Synthesis, meaning that we have two smaller substrates. When it binds to an enzyme, it will become a larger product. We call it synthesis. An example will be, well, we have amino acid, and then when you connect two amino acid, the covalent bond between the two amino acids, what do we call that? We call it a peptide bond. Peptide bond, so when two amino acids connect together to form a larger amino acid chain, what do we call this reaction? We call it synthesis. But it's an example of synthesis. What is the specific term for this reaction? Remember, it's called dehydration. So dehydration is a synthesis reaction, and hydrolysis is a degradation reaction. So everything we learned previously applied to a later lecture. Now I want to talk about induced fit model. Substrate and active site, they normally do not perfectly fit with each other, but when the substrate is being inserted into the active site, then the active site will change its shape. When the active site changes shape, in order to accommodate the substrate, we call this induced fit. So it's like, you call it induced fit. Originally, they are not the perfect shape. The shape does not fit, but the binding of substrate forces the protein or the active site on the enzyme to change its shape, and then we call it an induced fit. And then after the substrate is induced fit into the enzyme, what happens is that the enzyme can start to work on the substrate and then to speed up the reaction to form the product. So this is like the depiction. As you can see that the substrate has this shape, but originally the active site has a different shape. But after the substrate fit into the active site, actually the entire protein can change the shape, and then it will speed up the conversion of the substrate into product. Specificity. This is another characteristic of enzyme. Each enzyme, they will only speed up one reaction, and they will also work with one substrate. And because it's specific. So enzyme is specific to the substrate, and also they're specific to the reaction. And we usually name the enzyme according to their substrate or the reactant. For example, if the substrate or the reactant is lipid, then we call them lipase. So most of the enzyme, they end with ASE. So whenever you see a term that you have never learned before, but it ends with ASE, you can safely assume that it is an enzyme and it is a protein. So lipid is lipase, urea is urease, maltose is maltase. Where do you get maltose? Do you remember? Yes, it is the beer. You get maltose from beer. Nucleic acid, ribonuclease, lactose, lactase. So with enzyme, we can speed up the reaction much faster. An example is that hydrogen peroxide. If we have catalase, which is an enzyme in most of the living cell, catalase can speed up the breaking down of hydrogen peroxide 600,000 times faster. In our Bio3B lab that you are taking right now, most of you are taking right now, you will do an experiment using catalase from tomato juice. Using catalase from tomato juice to break down the hydrogen peroxide. And then you will measure the oxygen bubble with the ruler, how much oxygen bubble in the test tube with the ruler, as a way to quantify the rate of the reaction. The rate of enzymatic reaction will be influenced by all these factors. We will talk about each one of them right now. So, substrate concentration, enzyme activity, that means how fast the reaction, depends on how much substrate or how much reactant you have. If you have a lot of substrate, then the chances of enzyme catching the substrate will be very high. Then the maximum rate of this is achieved by the size of enzyme filled with continuous substrate. So when you have a lot of substrate, then the chances of enzyme catching the substrate will be very high. Then the reaction rate will be very high. It will increase. So the curve will look something like this. This will be the rate or reaction rate. This will be the substrate concentration. So in this direction you have more substrate, in this direction you have less substrate. It will be very fast, it will be very slow. So what happens is that for this, it is like something like this. This is the maximum temperature and pH. So enzyme will speed up the reaction as temperature increases. Well, it's very simple. When we heat up a reaction, what happens is that we'll supply more energy to the molecule, so that the enzyme and the substrate they will move faster because they are being heated up. When they move faster, of course, they have more chances to meet each other, and then of course the reaction will be faster in this case. So I just explained this, and then but temperature is not like, oh, the higher the temperature, you can speed up the reaction faster. No. If you continuously increase the temperature, after a certain point, the enzymatic reaction is actually start to slow down, and eventually it will actually stop. Why? The reason is that we call it a denature. Why is that? Enzyme is a protein. What happens when you cook eggs? The albumin is basically protein. Albumin will turn white and solid. Originally, the egg white, they are transparent and liquid, but after you heat it up, it becomes solid and turn it from transparent into white. It is also a denaturing process. Denature meaning that high temperature will change the structure of the protein. When the structure of the protein is changed, then what happens to the enzyme? The enzymatic activity will also be changed. So if we heat up the enzyme, at the beginning, of course, it will increase the reaction rate because the movement of the enzyme and the substrate will increase, so that the chances of the enzyme catching the substrate will also increase, then it will speed up the reaction. But if you continuously increasing the temperature, then the structure of the enzyme, which is also a protein, will start to change. When the structure of the enzyme starts to change, then the active site can no longer fit the substrate. When the active site can no longer fit the substrate, then it will slow down the reaction, or the reaction will not actually occur at all. It will slow, I'll just put it this way. When the active site of the enzyme is being changed because of the heat, then the substrate can no longer fit into the active site. Then the reaction converting the reactant into product cannot occur. Then there's no speed up because the reactant cannot fit into the active site. Then there will be no speeding up of the reaction. And decreasing temperature, decreasing enzymatic reaction, well, it's very simple. when we decrease the temperature, then the molecule of enzyme and the substrate, they will not move that fast. When they do not move that fast, the chances of enzyme catching the substrate will be lower. Then the chances of the substrate going into this active site of the enzyme will be lower. Then the reaction cannot be speed up. Then that's why it will slow down the chemical reaction. So the curve looks like this. It is the similar curve. So this curve is basically similar to this. The y-axis is the rate of reaction. X-axis, instead of the substrate concentration, it is the temperature. So you can see that, okay, as the temperature increases, then the reaction rate also increases, but up to a certain point, then if we continuously to increase the temperature, the reaction rate starts to drop because the high temperature will denature protein and changes the shape of the enzyme and also changes the shape of the active site. When the shape of the active site is changed, then the substrate can no longer fit into the active site, then there will be no speeding up of the reaction by the enzyme. One thing I want to point out is that what is the maximum reaction of this curve here? As you can see that it is approximately 37 degrees. That's why our body is also approximately 37 degrees, because many of the enzymes in our body, the optimum, the best temperature that they can work is 37 degrees. So ectothermic animals, that means the animal that change their body temperature according to the surrounding, like the lizard or snakes, their reaction rate is limited. Why? Because if their body temperature is only about, let's say 25 degrees, the normal room temperature, then as you can see that the reaction rate is not high. And if it is at room temperature and the reaction rate is not high, that's why you see that most of the snake or lizard, they don't move that much in the room temperature. But if we put a heat lamp, on top of the cage to warm them up, then you will see a small movement of the lizard or snake. For endothermic animal like mammals, our body temperature maintained at a fixed temperature, 37 degrees. So it doesn't matter whether we live in the North Pole or we live in the equator, animal can still move independent of the temperature of the surrounding. Next factor we talk about is pH. And each enzyme has an optimal pH to work on. It is very similar to the temperature. The reason is that enzyme is protein. pH can also change the shape of the protein. If it is too acidic or if it's too basic, the shape of the protein will be changed. We will give you an example of trypsin and pepsin. Trypsin and pepsin, they are enzymes, but they do not end with ASE. So some of the enzymes, they do not end with ASE. These two enzymes, they both digest protein. They break down protein into amino acid. But you can see that the optimal temperature to work for each protein, they are different. Pepsin, optimal pH is at the acidic, like pH 2. But trypsin is a little bit basic, around pH 8 or 9. So can you imagine where we will find the enzyme pepsin? And where can you find enzyme trypsin in our body? Well, where would you have a highly acidic pH in our body? Yes, it is our stomach. Pepsin is actually an enzyme being secreted by our stomach, and it works the best in the low pH, that means in the acidic environment. On the other hand, trypsin is work on a relatively basic I would say, kind of neutral or a little bit basic environment. Where can we find trypsin? We can find trypsin in our small intestine. Because our body secrete bicarbonate ion. The pancreas secrete bicarbonate ion in the small intestine, making the small intestine relatively neutral or a little bit basic, and that's why the trypsin work the best in this pH. Enzyme activation. How enzyme, because think about it, enzymes speed up the reaction. So it's like you're stepping on the gas pedal of your car, it can accelerate. But we don't want everything to accelerate. Sometimes we want to slow down, or sometimes we want not only slow down, but we want to be able to control with the enzyme, whether or not, okay, it's like you have a car. You can start a car or you can turn off a car, the ignition key. So what is the ignition key for the enzyme? The ignition key of the enzyme is the phosphate group. So not all the enzymes are needed all the time. It's like you don't drive your car all the time, right? Your car doesn't need to be on all the time. So this is when your car is not on. This is when you put the ignition key into the enzyme to turn on the enzyme. So when your car started. So one way to do it is that by putting a phosphate group and then forming a covalent bond between the phosphate group and the enzyme, then you will change the structure of the enzyme. When the enzyme structure is being changed, then the active site is also changed. And when the active site is changed, then you will feed the substrate. In this case, then the protein is activated. When there's no phosphate group bind to it by the covalent bond, that means the shape of the active site does not fit to the substrate. When the active site does not fit of the substrate, then the protein is not active to speed up the reaction. And remember, this kinase enzyme ASE, it is also an enzyme. So activation of a protein or activation of an enzyme may require another enzyme. So kinase is an enzyme that can activate a protein or it can activate an enzyme. Kinase is the enzyme that will put the phosphate group to a protein or an enzyme. We call this reaction phosphorylation. Phosphorylation means that the product is gaining a phosphate group, or the reactant group. Meaning that kinase is putting a phosphate group to the reactant. Okay, the substrate or reactant is here, and then the kinase will put a phosphate group to this, and then you become the product here. So you can consider the phosphate group or phosphorylation is basically turning an enzyme on or off. Or you may consider it as an ignition key. When the ignition key is in the car, then the car is on. So I briefly talked about that, okay, enzyme is like hitting on the gas pedal to accelerate a reaction. When you have acceleration, you need to have a brake to brake the car, slow down the car. Enzyme inhibition is like the brake to slow down the car. So we'll talk about feedback inhibition here. What does it mean? One simplest form of enzyme inhibition is that the product that the enzymes, so enzyme binds to substrate, and then they form an enzyme substrate complex, and then the substrate will be converted into product. So after the product is formed, the product can actually go back to the active site of the enzyme. So it will occupy the active site of the enzyme, so that no substrate can bind to the active site of the enzyme. In this case, we call this a feedback inhibition. So this symbol is like inhibition. Arrow is like induce. This is like inhibit. So if the product can go back to bind the active site of the enzyme, and we call this process feedback inhibition. After the product occupies the active site of the enzyme, then the substrate can no longer bind to the active site of the enzyme. Then this step will not occur. When this step does not occur, that means in here it will be E, P, and then plus S. When we have EP plus S, then you will not have the reaction will not move forward. It will not move forward. Yeah. And it will remain EP plus S. This is what we call feedback inhibition. So what happens when the feedback inhibition occurs is when the product is used up. When the product is used up, then the product that occupies the active site of the enzyme will be removed. When the product in the active site is removed, so because we are using up the product, then another round of the reaction, the substrate can go back to the active site to bind the enzyme to start another round of the reaction.